Hello, everyone. Welcome to Birds Explored. So, this is episode 12, and today we are going to talk about Turacos. So, we have actually, we stopped publishing this video for a long time. So, then we are back, and we are going to continue to upload our videos under the Birds Explode series. So Peter is here. I'm going to take Peter to the studio. Hello,
Hello, Ku.
welcome back.
How are you?
I am fine. How are you doing? It's a long time.
Yeah, we took a little break there.
Yes, then we back, and we are going to talk about after common Grecals. We are going to talk about Turacos.
Yes, the last one we did was Common Grackles, right?
You look like excited.
Are you being facetious? Am I not excited? I'm excited, of course.
Yeah, I know you are excited. You look
Yeah,
excited.
yeah. Look
Yeah.
at me. I'm
Yeah.
excited.
Okay.
Turcos are
Okay,
fun.
so yeah, of course. I mean, it's really interesting bird group. So I'm gonna leave the first word to you, and you can start.
Yeah.
Then
So, yeah. So,
we have presentation, and I'm gonna take this presentation to the stage, but I'm gonna leave the first words to you.
yeah. Well, I could do a little brief introduction to the Turcos. So, Turcos are
Yeah.
a family, an order, and a family of African birds, the Musophagiformes, the Musophagidae. And you're somewhat of an expert
Right.
on this bird, having done some extensive studies um, on them using museum collections, study skins, and
Yes.
tissue. And I guess you have done some work with DNA that you'll tell us about. And
Right.
Turcos are really kind of interesting in that they're a diverse group, but they also have in the past been somewhat of a taxonomic mystery. I know when I talk to my students in my biology of birds classes, we get to the Turcos, I am usually telling them, like, well, for a long time, we didn't really know where they fit in taxonomically. Some people thought that they were close to the Hoatzin, the South American bird, um, or cuckoos, uh, and these kinds of things. But I guess you'll talk today about some of the findings you've gotten from your DNA work, and maybe we can get a little closer to the actual taxonomic position that Turcos actually occupy.
Well, yes, thank you so much for this introduction. Yeah, I finished, we finished actually. I started interest in Turacos in 2011, and after 10 years, 2020, we published our first publication. It was long monograph published by American Museum Novitates under the American Museum of Natural History. So, this publication, it was
Mm-hmm.
so excited because, we did really comprehensive work. We started at the beginning, well, we thought maybe it took a couple of years, then the project getting bigger and bigger and bigger. So, and never finish until 2020, but still we are continuing doing something else which is interested in which is related to Turacos. So it's a really wide separate group um, distributed in sub-Saharan Africa. So real green birds, I can say, very interesting, very handsome bird group. So I said the real green bird because one of the pigments, Turacoverdin, so it's really real green pigment because, well, as you know, the green color kind of, am I right? It's structural color, or how can I say,
Yeah,
in
the
bird?
most yeah in birds, most of it's reflected light. Yeah, green reflection.
Yeah, the
But the Turcos have an actual pigment. Yeah.
actual pigment Turacoverdin, which
Mhm.
is green pigment and real green bird, I can say. So So yeah, this is the I mean, short introduction, and we have the presentation here. If it's okay for you, I can take the
Yeah, let's take a
presentation
look.
page.
Yeah,
Yeah.
there you go.
So, well, this is one of the big Turaco species, great blue Turaco. We also did a phylogeographical, independent phylogeographic study on this species. But today we are going to talk about little smaller ones, the green ones. So, and we are gonna explore some biogeographical patterns of sub-Saharan Africa using Turacos. So, and I try to make some interesting title exploring I mean diversity phylogeographic insights from Turacos, which is related to sub-Saharan Africa and African biogeography. So actually, I did this presentation, the kind of different version of this presentation in British Ornithologist Club a couple of months ago in London. So they actually like this presentation, and the longer version of this presentation also published in uh, British Ornithologist Club YouTube channel. So I think if our audience wants to see something more about turacos and some other bird species, they can also see that presentation in YouTube. So yeah, this is the title, and I can move to second slides.
Can I just mention one thing quick?
Of course,
So,
please.
so our audience sees this illustration of this particular species of Turaco. If you just look at that bird, um, the phenotype, uh, you know, you could tell why some of the early ornithologists might have been confused about what its position might be. It's got a little bit of a hooked beak. It's got
Right,
toes, two toes facing forward and two toes facing back, which is called a zygodactyl position of the toes that woodpeckers have and cuckoos have. Um, and this bird has a particularly long tail and a crest. And what do you do with that? If you just look at it, it looks sort of like a hawk. It looks like a chicken. It looks like um, could be. It looks like a game bird, like a pheasant. So,
right.
just looking at it is just the beginning. And this work that you've done has sort of sorted a lot of this out. So,
Yeah,
um,
that so far, I mean, I think as far as I know, I think three different comprehensive publication already published. Two of them published in IBIS,
mhm,
British Ornithologist Union journal in England. So the first one, Reg Morrow, made a comprehensive review published in 1960s,
mhm,
sometimes in 1960s. So then the second publication about the position of these turacos based on the captive birds
mhm,
and published in 1999 in IBIS. And after that, um, I think some people published some individual species studies in ostrich. Then I think our study, uh, well, we used all the museum, well, not all of them, but I mean most of the museum specimens in AMNH and also field museum I will mention later. So we used all these skin specimens and also some tissue samples, but we know where they came from. Actually, the geographical location is very important for these kind of phylogeographic and biogeographic studies. So, and then we try to sample all the subspecies from all the green turacos. So, which is important. Another important point. Then I think we got the comprehensive review of the phylogeographic and phylogenetic in terms of phylogeographic and phylogenetic studies of turacos. So yeah, in this episode, I mean, the purpose of this episode introduces scope of the phylogeographic research on African avian diversity. The Turacos is important for sub-Saharan Africa, and I think we can say something in general for the African avian diversity. And the key topics in this episode, I'm gonna talk about a little bit phylogeography and species limits, Then I'm going to talk about the markers, DNA markers, mitochondrial and nuclear DNA markers. So in this age, I mean, everybody using the genomic data, but for this study, so we use the skin specimens, the ancient, actually we use the ancient DNA, so it's hard to get the complete genome from them. So we only use the mitochondrial DNA and also some nuclear markers. And we are going to talk about the African bird diversity, avian diversity, and the case in this episode is Turacos. I just want to mention at the beginning of this presentation. So so I said phylogeography, and I just try to explain what is phylogeography. So the study of historical process that may be responsible for the contemporary geographic distribution of individuals. So we usually focused on the DNA markers when we talk about phylogeography. And of course, phylogeography has two different components, phylogenetic and also geography. We focused on the DNA markers, try to get the phylogeny. Then we need to get another information from geography and try to understand actually geographical variation of genetic diversity. So we did the same thing for the common gracles, as you know. And for Turacos, I mean, we focused on all these green species, green Turacos. Traditionally, there are, I think, 18 different species in green Turacos. And then we try to sample each species, some of them really endemic to one of the top of the mountain. For example, Taurocco bannermani from Cameroon, and also Taurocco ruspolai from Ethiopia. Taurocco ruspolai only occurs in one mountain, top of one of the mountains, top of the, I mean, the mountain edge. So it's really endemic. So, and some of them has really wide separate distribution, like Taurocco porphyriorhopus or Guinea Turacco, Taurocco persa. And we try to focus each species, try to understand the genetic diversity using phylogeographical concept and make some inferences about evolutionary biology, because phylogeography helps explain the patterns of speciation and adaptation. And of course, we use some DNA sequencing as a genetic marker to explain the geographical variation of the genetic diversity in Turacos. So this is actually phylogeography, what phylogeography means. I just try to explain, but I put some extra information about Turacos. So if you want to say something, you can interrupt me, well, anytime.
Yeah, yeah. Well, let me just say something briefly now for the audience. Not everyone knows how we get the materials to analyze. And when you talked about, you used the term ancient DNA, and a lot
Yeah.
of people might think, well, ancient, it's tens of thousands of years ago, but anything that's sort of older than 50 years, 100 years, the DNA in a specimen can degrade, it breaks down, right? So
Yes. Yes.
um,
Yes.
you used the toe pads of some of the study skins that the museums have. So let's say you have a specimen of a Turaco, the study skin. It's just the skin stuffed with cotton, and it's got the data on
Right.
it.
Right.
Hopefully, it's
Right.
got
Right.
good location data. And let's say it was collected in 1960. So
Yeah.
that's not too long ago. So
No.
what we do is we take a little slice from the bottom of the foot. The bird has these
Right.
toes.
Right.
And you take a
Right.
sterile blade and you make a little wedge, and you take that wedge, and it's got the coating of the outside of the podotheca on the leg or the toes. And then there's cells underneath there that have DNA. And if the bird is only from 1960, it's relatively easy to get good DNA sequences from it. But let's say the bird's from 1890 or something further back, it's a little more
That's
difficult to get
correct.
all these sequences, right?
Really
So
ancient
that
DNA. Yeah.
yeah, right, right. So that's considered ancient DNA.
Yeah.
Um, but you've gotten pretty good at being able to get this material and getting good quality sequences that you can work with. So I think it's important that the audience knows that this is not necessarily fresh tissue, fresh blood from a bird that was
Right.
collected last week. Um, but there are techniques that have been developed over the past decade or so that really help in being able to get good material. And the other thing I wanted to say was that when we talked about the phylogeography of the common grackle in North America, the map was very different than what you see here, because the common grackle uh, contiguously occupies a very large range in North America. So there's a lot of hybridization and what we call symmetry, where the birds are together. But what you're going to be talking about here are birds that are separated by mountains or streams or elevation and that sort of thing. So they're not all just kind of smoothly from one place to another, so that the pattern will be different than something like a grackle.
Right, right.
Yeah.
Well, very, very useful information. Thank you so much. And I think we also should say, so all these specimens in Natural History Museum collections makes the natural history very significant, because all these specimens kind of the component of the natural history as a component of the history, the
Sure.
part of the history. So it's very important, and it's very, very important to protect them, very important to use them to get some useful information about natural history, because then we can easily understand all these biodiversity patterns, all these different biogeographical patterns, and try to understand the past. It is very important to predict the future.
Hmm.
So, and we in this study and all these studies, including common grackle and other studies, we try to use this perspective.
Hmm.
So I just want to say, because nowadays, natural history is very important, and I think nowadays some people try to take the history out of the natural history. So that's why I just want to say, I
Hmm.
just want to put this concept.
Yeah, I agree.
And yeah, yeah, we focused on this group. Um, I mean, I worked with George Baraklov and Jeff Groth, and we focused on the Turacos. And 2020, we published this monograph in American Museum Novitates. I was really excited. I'm still excited. I really like this publication. So it's very nice contribution to understand some different patterns about African biogeography, African bird biogeography. So we try to, understand all these genetic mysteries of Africa's this colorful bird group. So yeah, this is the this is the handsome bird, one of the species in green Turacos. So we try to get the topet from all these species, all these individuals that we used in this study from the different species. Then we try to get the DNA from topets. So we probably sampled over 500 specimens, individuals, but I think we got only 414 individuals get success. We got success from the getting the DNA from 414 or 419. I couldn't remember, but if the audience look at the paper, it's open access in Bio One, so they can easily see, and it's over 400 individuals. We got success to get the successful DNA. Then we use the first mitochondrial DNA. Mitochondrial DNA is very important DNA marker, because it's easy to get when we compare the nuclear DNA uh, genes. So because in that time, 10 years ago, um, the genomic studies just started, and we didn't get enough money, and we didn't get enough technology to get the DNA from topets using the next generation sequencing techniques. Then we use the traditional techniques, and we got the individual gene. The first we started mitochondrial DNA, and we got this haplotype network. As the audience can see and as we can see here, there are lots of different colors, and each color represents different DNA sequences. And as you can see, Peter, I mean, there are lots of genetic diversity here because each color shows different DNA sequences, which is indicated different genetic structure and different genetic diversity within each species. And here we have 18 different species and different populations, and we understand some of the different populations is really different than we can talk about the different species in one species. For example, in one species there are some subspecies, but based on the DNA results, we decided these subspecies is a different species. I'm going to explain later in the different slides, so the audience can easily understand after that. But here, I just want to show there are lots of different DNA sequences, and there are lots of genetic diversity in this group. So, for example, we can focus on this shading area, the orange shading area. There are different species over there, and also this species, I mean, far from the main group. Main group is here, and there are lots of, in little black dots indicates the mutations. There are lots of mutations accumulated over the time. So these species looks like similar with the green Turacos, other green Turacos, but they got lots of different mutations over time historically. Then they got different, I mean, clades. Then they are different than all these, I mean, main green Turacos, but they are still green. But there are something happening in the group. So it indicates there's a long history in this group. Then we can try to explain some individual patterns, and then we can come to the general picture after that. So this species, Turacos distributes in the region of the east part of Africa. It's very widespread distribution. Sorry. Here in the east part of Africa. So there's, I mean, there's a line between black and white genetic structure. This line indicates the Zambezi River. So the Zambezi River is one of the important geographical barrier in east part of Africa. So when we look at the north part of Zambezi River, we can see the black dots is common. And in south part of the Zambezi River, we can see some white dots, white genetic structure, white DNA, or, I mean, we can say that let's say the white genetic structure. But there are one location, black and white, I mean, meet and they got together. But mostly the black haplotypes, north part of Zambezi occurs north part of Zambezi River, and white haplotypes occurs the south part of Zambezi River. And if we look at the haplotype network in the middle of the figure, in the bottom side of the middle of the figure, so between white and black haplotypes, there are something happening. They just they look like the mix a little bit. But if you look at the subspecies definition on the left-hand side of the slide, so the one subspecies has brownish breasts, another subspecies has no brownish breast. This is really recognizable morphological differences. And brownish breast has different genetic structure, like white or black, and the other one has different genetic structure, and these morphological differences looks like a consistent based on the specimens in the collection, because we use the AMNH specimens, we also use the field museum specimens, so we always see really consistent morphological differences between these two subspecies, and the genetic diversity looks like a different. Then we decided to say there are two different species in this group. There are little historical hybridization between these two subspecies, but all these evidences, like geographical barrier and the haplotypes distribution, indicates and supports this decision. And also we got some nuclear DNA results, and nuclear DNA results also supports this genetic structure, because this genetic structure mostly based on the mtDNA results, mitochondrial DNA results.
And
This is one
and
in example. Yes.
and we're sure this is not sexual dimorphism. The males have the collar and the
No,
females don't.
in Turacos, male and female usually alike. There is no differences between male and females. So, and then in the breeding time, also male and female, I mean, just try to help to clutch to raise the, chickens actually after the hatching period. So, and there is no sexual dimorphism between male and females also.
Okay. And the specimens are identified as far as males and females as well.
Right.
Right.
And then if we look at the label information, we were really sure they are male and female. Yep. So the another interesting example, west part of Africa, there are some green distribution. You can see, the very west part of Africa and the east and west of Africa. So there is a big gap between east and west, which is called the Dahomey gap.
Mm-hmm.
So if you look at the west part of Africa and eastern part of the west part of Africa, if you look these two distribution, you can easily see, if we go there, we can easily see, rainforest there. And then some Turaco species use rainforest. And between these two distributions, there is one Dahomey gap, which is characterized by savanna. There is no rainforest.
Mm.
And then we got the samples for the yellow-billed Turaco, Tauraco macrorhynchus. The scientific name is Tauraco macrorhynchus.
Mm.
And if we look at the specimens, one of the allopathic distribution indicates the one subspecies, and this subspecies, had red crest, little differences, and another one black crest. This one also distributes in the eastern part of west part of Africa. So this subspecies actually has a geographical barrier between these subspecies. The geographical barrier is the Dahomey gap.
Mm-hmm.
So and these subspecies characterized, as I said, red crest and black crest. And we got the DNA. We look at the DNA differences between these subspecies, and we found, as the slide shows, we found some private haplotypes occurs in west, and another private haplotypes occurs in east, and there is a big differences between these haplogroups. And these little morphological differences match with the haplotype differences, mitochondrial DNA results. And also, nuclear DNA results also support this. And then we can say there are not only the subspecies, probably there are clear two phylogenetic species, because they use some allopathic distribution.
Mm.
All these DNA markers shows there is no clear genetic gene flow, and there is a clear genetic differences between them, which shows no gene flow between them. Then morphology supports these results. Then we said, in our study, there are two different species, not the subspecies. So, which is very interesting. But if we look at the another group, which is purple here, and again, the male and female looks like alike,
Mm-hmm.
and there is no subspecies, they look like a monotypic. So, and there is a really, I mean, big distribution in the middle of the Africa, and the genus name different, Musophaga rosei, and we got, I mean, north part of the distribution range. We got the samples from the north part of distribution range, and we got the samples from the west part, west and east part of distribution range. And if you look at the haplotype network between the DNA, there is only one mutational differences, and there is no haplogroup like Taurocomacrorhynchus. So it's a wide distribution range, really wide separate species, and there is no genetic differentiation within species, which is also interesting results. Some Turacos species shows really clear differences based on the DNA markers. Some of them has really wide separate distribution, and we can't see any difference in the DNA.
Mm-hmm.
So this is interesting. I just want to show these two conflict in one slide. So yeah, another important example from middle of the Congo. Not middle of the Congo, middle of the Africa, which is Congo. So in Congo, there is one Turaco species, really wide separate distribution, which is Tauraco shuetai, black-billed Turaco. Well, as you can see in the drawing, one subspecies shows clear green back and wing.
Mm-hmm.
One subspecies shows really purple tail, back, and purple wings.
Mm-hmm.
So, so the people says, I mean, one species and two different subspecies, Tauraco shuetai and Tauraco shuetai emini. They are the scientific names. So please don't forget this example, because I'm going to come back this example when I show the big phylogeny. So we got the DNA again, the same thing like the previous slide. And if you look at the middle of the haplotype network, middle of the slides, so Eminia and Shuetai has really, I mean, lots of mutations between them, and we couldn't really find the geographical barrier in the middle of the Congo. We don't know what happened. But they are completely different based on DNA, and they are completely different based on the morphology. But people says they are subspecies.
Mm-hmm.
Until we got the complete phylogeny, so we said, okay, different subspecies, maybe they share the common ancestor, and they are different. There are some different phylogenetic species. But I'm going to show you some interesting results a little later. Then we can move to some small distribution. Some Turacos has small distribution when we compare the other big distributions, like Tauraco heart lobby, because Tauraco heart lobby only distributes in Kenya,
Mm-hmm.
northern Tanzania, and east part of Uganda.
Mm-hmm.
And this species only use mountain tops, like Kilimanjaro, like Usambara Mountains, like Elgon Mountains, and like Mount Kenya. So we have in AMNH very nice, I mean, set of collection for Tauraco heart lobby collected by Chapin. I mean, Chapin made a really nice exhibition in Africa, as you know, and he collected very nice set of Turacos and other species. So we try to get the, I mean, DNA from these species, and people accept this species monotypic. There is no subspecies in this group. All the populations looks like the same. I mean, like this figure. I mean, there is no little differences between different populations. I mean, if you look at the Kilimanjaro and if you look at the Algon, you can't see any differences. So we got the DNA, and then it was so surprising, and we got completely different mutations, different DNA sequences from each population. I mean, if you look at the Usambara, there's completely different genetic structure, really isolated population there. And Kilimanjaro, different genetic structure, really isolated population. Algon the same. North part of the distribution range is same. They are all mountains. There are some gene flow in the middle of the Kenya. There are some mountains like Mount Kenya and some hills, and there's some common distribution in the middle of the Kenya, and there are some gene flow between populations. But there is no morphological differences. You can't see any differences when you look at the specimens. They are all the same. Then we got to this haplotype network, and Uganda actually monophyletic to others, and also Kilimanjaro and Usambara are also monophyletic. So, which indicates something very interesting, but we need to get some more evidence to make a clear discussion. Something like a cryptic species, but we only got little evidence based on the DNA, and I just want to get more and more evidence like song data. But I already looked at the morphometric measurements. I couldn't see any differences based on the morphometry. But we saw some significant and real and very important differences in DNA. So it is small distribution. Remember Musafaga rosy, there is a huge distribution. There is no differentiation within species. There is a little distribution when we compare the Musafaga rosy. There are lots of different mutations occurs in one species, which is very interesting. So then this is the gene trees. I mean, we got the mtDNA, we got the one intron from nuclear DNA, and we got all the subspecies, and we try to make some gene trees and try to compare these gene trees. And these two actually different markers not completely, but mostly match each other. There are some disconcordance like that place and then this place, and we try to explain all these disconcordance our paper. If the audience really, interested in the discussion about this, they can see the paper. So, but remember Turacos shuetai, because I just want to remind you, well, the one subspecies, Turacos shuetai, I think here, based on the mtDNA and also aconitase, it, located here, but if you look at the amini, where is amini? Yeah, the amini located here.
Down at the bottom, middle, sort of.
Yeah, here, and then shuetai is here. So people accept these two people accept these two populations occurs in one species, and there are two different subspecies, but they don't share the common ancestor. So we can't say they are subspecies. They are completely different species. Not only phylogenetic species, also probably the biological species. So they are not sharing the common ancestor. So this is one gene, this is another gene, but we also got one exon, reg1, and we made a species tree, not the gene tree. We got all these genes together, and we got over 4,000 base pairs from different markers, from different evolutionary rates. Then we got big phylogenetic tree like this one. Look at the amini. Amini is here, shuetai is here. The common ancestor for shuetai located here. Common ancestor for amini probably located somewhere here. They have the different common ancestors. They are not diversified from the one that they are not sharing the common ancestors. So we can't say that there are different subspecies. So we got some really nice support for example, the Porphyriorhopus. 100%, two different subspecies, located in sharing its common ancestor, but two different subspecies, clearly you can easily see they are phylospecies. So, well, there are some gene flow between them, but if you think about the phylogenetic species, it's not really important. So the inbreeding is not really important. So breeding is not important. I mean, there are two different phylospecies, and when we look at this phylogeny, we can easily talk about, well, traditionally 18 species described, but I think after this phylogenetic study, we elevated some subspecies at species level based on the phylogenetic species concept, and we can talk about the 25 different species in green Turacos, which is really interesting. And there are lots of endemism pattern described using, I mean, all these genes and described in this study. So, yeah, I'm coming to the end, but our study integrates, I mean, mtDNA and nuclear DNA data to refine the phylogenetic relationships within Turacos, confirming the distinct lineage of major groups, revealing new insights, of course, into the evolutionary paths. And we have identified significant genetic diversity and structuring within Turacos across montane and lowland forests of Africa. It's important for the biogeography of Africa and suggesting historical climate events have greatly influenced their current distribution and diversity. And unexpected genetic breaks in Turacos without clear phenotypic or habitat differences. I mean, challenge traditional views on their speciation. The positioning of also, I mean, I just want to say the copper mines suggest a significant but yet overlooked impact of this genetic differentiation, because at the beginning we talked about turacoverdin and turacin. I mean, these two pigments need some copper, and these birds, these birds, well, eating the, I mean, some fruits, not insects, not other animals, based on, well, they are really depending on the vegetation, and then vegetation gets some copper from probably the soil, and they really need to get the copper for their feather coloration or their feather pigment stuff. So the this is overlooked impact, but I think the copper mines maybe suggest something, because for the Shuetai, for example, I couldn't see any significant break between two different subspecies or two different things in species. So maybe these kind of signal shows something us for the evolutionary, to understand the evolutionary break. And it's highlighting the complex role of the human activity and subtle environmental factors in evolutionary dynamics. And in the summary, well, our results indicate that there is a substantial amount of previously unrecognized genetic diversity in African birds, particularly in montane and lowland forest areas. These results indicate that we need to reevaluate the existing avian biogeographic zones and historical distribution models across Africa. So the Turacos really helping us to understand the biogeographical pattern in Africa. And also I just add some interesting stuff, culture in aviculture, if you think about Turacos, because all these native people in Africa use some bird feathers, well, as you know. But their red pigment is very important, and their red pigment, tracin, has inspired myths like stories of bleeding wings in the rain. Because, well, if you put the red feather in the water, the color is just start going out from the feather because of
I've
the
done
tracin.
I've done that in class as a demonstration.
Right, right.
Yeah, taking the primary feather, put it in
Yeah,
water, and moved it around, and it
yeah.
bleeds into the
It
solution.
only occurs in that bird group. So, and in some cultures, Turacos symbolize the vitality and courage, adding depth and their value and the agriculture. So it is,
Hmm.
which is interesting. And also, the colorful Turaco feathers are commonly found on the ceremonial costumes of the forest people of Central Africa. So they use um, Turacos for the ceremonies. So that it's important for the people, well, native people, not all the people, but native
Mm.
people, using using these feathers, making some masks
Mm.
also. Well, thank you so much. This is my presentation. I just try to introduce the Turacos using some genetic data, but maybe something technical. But I think I can explain something about the diversity in Turacos and diversity in avian groups in Africa.
Yeah, well, sure. There's some technical aspects to it, but the maps with the genetic networks on there are pretty interesting to see. I mean, right away, anybody can see that there's this great diversity, but then also the differences in the altitudinal forms.
Right.
That you know they were all pretty much the same in one place, and then you go to another highland area, they're the same there. But then as you get down into the lower elevations, there seems to be more of a pattern there. So we're talking about isolation, right?
Yes.
Evolution,
Yeah.
yeah, evolution taking place. These isolated populations.
Like an allopatric speciation. Yeah.
Yeah, and we also mentioned maybe doing some work with vocalizations, and
Right.
yeah, so they're not songbirds.
No. No.
So they I don't know what they sound like. They have sort of a call type or a basic sounds that they make.
Well, they actually, um, if we get in the rainforest, they make the, I mean, really, they really characterize the rainforest with song. Their song look like, if we, if the audience can go to Zenocanto or other
Mm-hmm.
sources
Mm-hmm.
like eBird or Birds of the World online platform, they can easily see, they can easily, I mean, listen the
Mm-hmm.
some sonograms from Turacos. Their song looks like a barking dog.
Okay, so
They
it's
are, they look really handsome birds, but their songs is really weird.
yeah, well, it's not like a melodious kind of
No, no,
tune.
not melodious. It's kind of like a barking dog,
Yeah.
and they always use the higher part of the canopy.
Uh huh.
It's hard to see in the rainforest because
Right.
the rainforest is dark.
Yeah.
So when you get in the rainforest, you can easily, I mean, see hear some barking dogs, like a mystic song. So kind of a
Right.
horror film, horror movie. So they are all from the Turacos, I think,
And
but they are
I imagine,
always using higher level of the canopy, and it's hard to catch them. It's hard to
yeah,
see them.
yeah. And I imagine it carries a bit. Um,
Right, right,
yeah, something like in South America, something like a bellbird,
right. Yeah, yeah,
something like
that's true.
that. Yeah, it'll be interesting. I don't believe I've ever heard them, but I guess you could find them on YouTube or
Right.
Canto.
There are lots of different songs from different species. Not very good quality, but in Zenocanto, people
But
can people can listen.
that simplified vocalization though is relatively easy to analyze. Then you don't have a lot of noise. You don't have a lot
Right.
of other
Yeah.
things going on. So a
Right.
particular note, just means one thing. Um,
That's true.
and there, do the males do they lek, or is there a single male that dances? The
No,
mating
I
system
think
like?
the mating system is that they are mostly monogams, not the
Mm-hmm.
polygyn birds. So
Mm-hmm.
there is not like polygyny,
Mm-hmm.
or there is not any other special mating system. They are like monogam bird. They are native bird,
Mm-hmm.
not migratory, completely
Right.
resident and
Sure.
endemic. So I think the sex ratio almost 50-50. So
But is there an
I'm sure,
elaborate?
as far as I know, that I am sure there are some competition going on,
Yeah.
um, but I think, yeah, the little sexual dimorphism also plays an important role
And
when we
size.
see
Yeah.
animals.
Yeah.
The size, and
Yeah.
also, of course, the size is
Yeah.
different. But well, I try to show you so that some little differences between subspecies.
Yeah.
I think some sexual selection maybe at some point important for Turacos.
Yeah. Is there an elaborate courtship display? Is it just the male that does something and the female just kind of watches? Do
As
you
far as
know?
I know, the male does something, and then the female picked one male. Yeah.
Hmm. Well, that'd be interesting to look into because I don't know anything about it. But
Right, right.
you know,
Well,
and
there's a
they
one big monograph published, I mean, by Forshaw and Cooper.
right,
Well, I have that book. I mean, it's very interesting, and they
yeah,
try to
tells you everything in
review
there.
all the information. It's not a recent book. I mean, published
Yeah,
in 1990s, I think.
yeah, but
But
it's
really
comprehensive.
handsome, really very nice drawings in it,
Right.
and there are really good information. Also, they got some measurements from Turacos. So yeah, this is the monograph so far published and explain all this breeding, habitat, and distribution
Yeah,
information.
so I guess I think Cooper's the artist, right?
Yes,
And
yes, yeah,
Forshow's the ornithologist.
right.
Um, and what are they eating? They're
For the,
frugivorous. Yeah,
for givers, yeah, definitely.
yeah,
They
right.
are just eating like, well, some common name says banana eater
Yeah,
for them, so they are really eating just the fruits.
yeah. Well, they must feed on some insects for protein and maybe even
Oh yeah,
small
of
vertebrates. Yeah,
course, but not really, not
right.
generally,
Hmm.
not usually.
And they're feeding on the ground, or
No,
in
they're
the trees
feeding on the.
potentially.
I mean, no, they only use the trees and the
Yeah. Oh,
canopy.
okay. All right. So they're in the canopy.
Yeah,
So
they never. It's hard to see them.
right. Okay. So
Well,
see, I'm more.
I think some gray go away birds, but not all of them the green. So some
Right.
of them like the bigger size, like great blue turaco. It's
Right.
completely different,
Right.
but it's part of the group. But the
Yeah.
other parts, the gray turacos, they're all gray.
Yeah.
Also, they are part of the turacos, but it's far. So, but that ones, the gray ones, I think it's easy to see them because they usually use the savanna, not the rainforest.
Yeah.
Like I forget the common name. I mean, gray goyle bird,
Gray
gray
gold.
goyle,
That's a
which
big
is very
bird, right?
the big bird and
Yeah.
bigger than the green one.
Yeah.
The most big one, the really big one, the great blue turaco,
Right. Right.
great blue turaco. So it's really big one. So the
Yeah.
gray one bigger than green turacos, and they mostly use the savanna, and it's probably easy to see them on the field
Hmm.
work in the field.
Yeah. Yeah. That's where my bias is. I'm thinking about the bigger ones, and so I think about them being more terrestrial, but they're
Right,
really
right,
not
right.
necessarily. Yeah. And they're building stick
Oh
nests in the
yeah,
tree.
they just try to find the correct branch like
Hmm.
this, make a platform,
Mm.
nests, and male and female, I mean, help each other to make the
Mm.
nests, as far as I know.
Mm. Yeah. Yeah. I'd like to revisit that. I haven't looked into their breeding biology in a
Right.
long
I mean,
time.
these Coopers, I mean,
Forshaw and Cooper.
Forshaw and Cooper monograph really helping. I mean,
And not
we try to review some information, but not really the breeding information.
right.
So
Sure.
we got some morphological information. We got some, well, mostly DNA stuff, but we also check the measurements.
Sure.
So that's it.
And I imagine not too many eggs. Probably.
No,
Probably.
2 eggs. I think 3
Yeah.
eggs.
Yeah. That makes sense.
Yeah, not too many eggs. No.
Yeah.
Yeah.
Okay. Well, that was uh, that was very interesting. It's another one of these really cool groups of birds. They
And
are bred.
yeah, I should also say, I mean, the common, well, when you go to the uh, basal of the tree,
Hmm.
phylogenetic tree, so the people used to accept Huatzin from South
Right.
America, share the common ancestor, all the Turacos.
Right.
So, but I think there are some recent paleontological studies published in BMC Evolutionary Biology. They found some fossils in North America.
Right.
and they got some measurements, they got some review of this fossil, and then people understand, all authors and researchers understand the dead fossil found in North America related to the Turacos.
Mm-hmm.
And then Turacos only use the forests. They
Right.
depended on the trees. And then they said long, 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 long time ago,
Mm-hmm.
also the North America has really some forests,
Sure.
like a
Yeah.
rainforest,
Right. Sure.
something
Right.
like that. Then all these continental drift and everything, they are differentiated. So, and it's some other studies published in Nature, and Rick Prum, and also the other authors, other researchers published in Nature, not a couple of years ago, maybe 10 years ago. So there's a big poster on the lab in the sixth floor.
Mm-hmm.
You can check in the molecular lab in AMNH, sixth floor. So they found some evidence Otis tarda, the bastard,
Right.
shared the common ancestor with Turacos.
Mm-hmm.
Because of this, rooting of these phylogenetic trees, we used cuckoos, Otis tarda,
Mm-hmm.
as an outgroup
Mm-hmm.
to get the correct routing for our tree, and we cited all these papers.
But not the whole Hudson. So
I
so
think
it
we
looks
also
like.
use Watson. Yeah, I think, yeah, we put,
Oh,
we
okay.
mostly we put the cuckoo and Otistarda, not the Watson, I think. Yeah.
Right, because it looks like maybe if these North American fossils are legitimate, then it might be the origins of the Turcos in the northern regions, and not in the southern or the South American rainforest regions.
No, no,
Yeah.
no.
Mm-hmm.
Yeah.
Yeah. Yeah.
So,
And
yeah,
that's counterintuitive too, with the way that they look. The Turcos don't look
right.
like bustards, you
No,
know.
no, the Buster is completely different. We should, yeah, we should move to probably the Watson, the next one, next
Yeah, in the
episode,
next
and then
episode. Yeah.
the Watson, and then after Turacos, maybe, and after that, we should also discuss bastards,
Okay, let's do that. The
including
next two.
Otistarda. Yeah, yeah, next
Right,
to,
that makes sense. So folks
yeah,
remember the Turcos, or look at it again, and then the
right,
next time we'll do the Watson, which what whether it's related to Turcos or not is its own fascinating weird thing. The Opisthocomidae is the
right.
family, and they got weird digestive systems and all kinds of neat stuff. So next time let's do that, and then the bustards, the Otididae, after that.
Yeah,
Um, Again, big diverse. Like the Watson is not diverse at all. It's one thing, but then the
right.
bustards, of
That's good.
course, you got big ones and little ones and all kinds of neat stuff for that. So that's good. All right. Well, hopefully everybody will take a listen and a look at the presentation on Turcos, and take a look at your papers if they're inclined. I know a lot of our audience, I guess, is mixed. Some people are just interested in birds kind of casually, and then some
Right,
people are maybe more scholars and are interested in reading the papers. And people are welcome to comment. We don't get a lot of comments on these, but
no, no.
if we've over,
Hopefully, we can get comments. Yeah,
yeah, if we've overlooked something or made a mistake, or that's fine.
of
Point those
course.
things out. Just have a discussion about this stuff. So
Right.
good. All right. Well, Utku.
Thank you so much, Peter.
No,
So it
thank
is
you.
nice episode,
All right.
and we should just wish. I just want to remind the people also. I mean, the episode 11 we talked about common grackles,
Mm-hmm.
and now we talked about Turacos. Common grackles is the PhD of the Peter, and we try to discuss one of the migratory birds in terms of the phylogeography and the morphological variation. And Turacos, I mean, I spend lots of time with this species, and this is completely native species, not migratory. And
Right.
if you look at the two different papers in American Museum Novitates, the Turaco paper and Biological Journal of Linnean Society published the common grackle paper,
Mm-hmm.
and they can just make a comparison between two different behavior and two different DNA results.
Yeah, and of course, there's all these subtle ecological things that we probably have no clue about that are
Right.
driving a lot of what's going on.
Right.
So, but we do the best we can with the tools that we have, and this is what ornithologists do.
Of course. Yeah.
So if you're interested in the kind of work that we do, this is part of it. So, all right,
Yeah.
good.
We learned something, but we also got some ignorance.
Oh, well, you took more
Yeah.
questions. Yeah, yeah, of
More
course.
questions after
Sure.
that.
Well,
Anyway,
that's the way it's
thank you
supposed
so
to be.
much. Thanks
All
a
right.
lot.
I'll talk to you later.
And yeah, talk to you later, and let's try to meet in a short time for the episode 13.
Okay.
Bye.
All right.